Have you ever wondered how we sequence DNA? Well, let's just take a quick look at DNA sequencing. So we're going to break down DNA sequencing into 3 different steps. So the first step is you take the sample of DNA that you're interested in sequencing, and you basically use PCR to amplify the sample. So by using PCR in order to amplify the sample, you're able to generate lots and lots of DNA fragments. So the next thing that you do is normally in PCR you have to add nucleotides. You have to give the growing strand the substrate from which it can grow. So normally you add in regular deoxynucleotides, and those look something like this. You've got an OH group here, you've got an H group here, you have a base, and then you've got carbon group, and oxygen, hydrogen. So this is what a normal nucleotide looks like. But interspersed in the PCR, what you also want to add is you want to add in something known as a dideoxynucleotide. So a dideoxynucleotide looks something like this. It's basically exactly the same thing, but it only has a hydrogen here. So this oxygen is removed. And what that basically does is if this dideoxynucleotide, we can abbreviate DDNTP, if this incorporates into the growing strand, since there's no oxygen group here, the strand can no longer elongate. So you basically have termination of strand elongation as soon as this DDNTP incorporates. So what you can do is you can actually fluorescently label the different dideoxynucleotides. So for example, we have 4 different options. So we can label all the G's blue, we can label all the A's red, all the T's green, and all the C's orange. And so basically what you have is you have these dideoxynucleotides with different fluorescent labels getting incorporated into the growing strand. And since PCR is able to amplify, create millions and millions of DNA fragments, you can basically, what you can do is you'll have strands of different lengths. So let's just kind of look at an example. So let's imagine that we've got nucleotide being incorporated here, a regular nucleotide, and then another one incorporated here, and then another one, and then just randomly, all of a sudden, we have a dideoxynucleotide being incorporated here. And this would stop elongation of the strand. So you'd have a DNA strand, that's just 4 nucleotides long. And after another round of PCR, what we might have is 1, 2, 3, 4, 5, 6. It's just growing, it's growing, it's growing, and then all of a sudden, whoa, what happened? You got a dideoxynucleotide being incorporated. And so basically you just do this, and after you've got millions of samples, you'll eventually be able to have something that looks like this. You'll have maybe just one regular nucleotide and you've got a dideoxynucleotide incorporated, or you might have maybe, let's say, 2 of them. So you'll have 2, and then you've got let's use this color. So you've got a dideoxy. So what you can basically do is you can see that you have strands and they're elongating, and different strands are terminated at different points by a dideoxynucleotide. And so basically the next step is you use gel electrophoresis in order to separate the strands by size. So when you run all the different fragments on a gel, it'll separate them by size. And then you can just have a computer go in and analyze all the fluorescent labels. So if it sees here that you've got this blue fluorescent light, then it knows that the second nucleotide in the sequence is a G. So it'll say G. And then it'll look here and it'll say, okay, well this is a C. It'll look here, it'll say we have another G, and so on and so forth. And basically the computer is able to, by reading these fluorescent labels, these fluorescent tags, it's able to give you a DNA sequence. And so this is basically an overview of how DNA sequencing works.